Botanicurus is a plant that contains an enzyme that allows scientists to cure cancer. Scientists often conduct various tests to check for genetic relationships. In this scenario, you are the scientist conducting a test to find what species is closest to Botanicurus. The reason you are conducting this test is because Botanicurus contains an enzyme that can cure cancer. So you must find the closest related species that also contains this enzyme, since Botanicurus is going extinct. Now, it's really important to remember that this is a simulation, so it's not a real plant and doesn't really cure cancer, but it's a simulation. Step 1. Read the brief introduction on page 1. The introduction is from here to here. You must read that paragraph in order to complete step 2, which is to identify the objective of the lab. Once you are done with step 1 and step 2, define some key terms that will help you understand this lab with more clarity. Define structure and explain why structural similarities between organisms point to common ancestry. For step 3, you must complete the first column of the table on page 8. So we turn to page 8. Please do that now. If you look at the table on page 8, you will see that along the rows are first Botanicurus, then 3 other species that resemble Botanicurus. Those are X, Y, and Z. If you look along the columns of the chart, you will see that there are various tests that you will be performing in order to find which one of these species are closest to Botanicurus. The first test that you will complete is the structural similarities between the plants. That means that you will identify and describe the structures of each plant. So you will start with Botanicurus, and we will identify the structures that you see in this plant. For example, one of the things that I can note is that the petals are purple and yellow, and that there are black rays coming out from the middle of the flower. You can discuss the type of leaves, the shapes of leaves, the size of the plant. You can discuss the relative amount of leaves the plants have and the numbers of petals each flower has. After you are done describing Botanicurus, you will describe X and then Y and Z in the same way. Step 4. Complete the second column of the table on page 8 by observing the varieties of seeds from the plants. So now we go to column 2 and we do almost the same thing as we did with column 1, but we use the seeds for comparison. These are the seeds of the plants. This is the seed for Botanicurus. X, Y, and Z. Some of the things that you can speak about when describing the seeds is the shape, the color, the size, the patterns, the relative thickness, and so on and so forth. At this point, we are going to observe the plant stems, cross sections of the plant stems under a microscope. So we take Botanicurus first, slide it into our microscope beneath the microscope clips, right underneath the clips. We identify, some of the slides will have a green tint to them, so we identify where it is, And we want to put that spot in the middle of the microscope so that we can find it very easily. And then we want to start with the lowest possible magnification. In my microscope, it's 4. That's the lowest. It's usually the smallest objective. I click that into place directly over my slide. And then I'm going to use the coarse adjustment, which is the big knob over here, the coarse adjustment, until I can identify it, center it, and focus on it. Once I've done that, and it's visible, I am going to now change the magnification slightly by moving the objective to the next highest magnification. Now that it's at a higher magnification, I can use the fine adjustment, which is the long knob, adjust it until it's perfect and clear. And once I have it perfect and clear, I can make it bigger yet again. And then I can magnify it until it's clear. And then I should be able to see the plant in the highest possible magnification. That's how slides are used in the microscope. And then you will do the same process for X, Y, and Z. Once you are done, your data from this goes on underneath the microscope column on page 8. You can draw directly what you see inside the microscope onto that column for each one of these, or you can write what you see. Thank you.
At this point, you will have to go backwards in the lab to page 2 in order to form a hypothesis based on the structural components alone. The first part of the question on page 2 asks you to pick a species that is most likely to produce curol. The second part of this question asks you to support your hypothesis with evidence from your data table of structural comparisons.
You are now ready to complete step 7. Step 7 involves a paper chromatography. In order to complete this, please read the little information about paper chromatography and explain what paper chromatography is used for. Then also explain in what way paper chromatography will help you to identify the most closely related plant species. Once you have answered those 2 questions, you are ready to move on to the paper chromatography experiment portion of the lab. To do that, you will need a filter paper, which most often comes in a circular shape. Cut it into a square so it's easy to use. Put a line about 1 centimeter above the bottom of the paper. Then you have your starting line. In this starting line, you will mark 4 dots. So 1 will be here, 1 here. Try to make it evenly spaced as possible. And underneath the dots, you want to indicate what you're going to put in each dot. So you're going to put a pigment in each one of these dots, but you want to indicate whether it's Botanicurus. I'm going to write BC for Botanicurus. I'm going to write X for the next one, Y, and Z. And when I am ready, I'm going to use, of course, a different pipette for each one so the pigments don't get mixed up. A little bit background. These pigments are pigments that are taken from each of the plant species, and we are comparing these pigments to find which plant species may be the most closely related. So Botanicurus pigment, 1 drop right over here. Not too much because I don't want it to spread too much in the paper. So same thing, 1 drop on X, and squeeze my pipette, insert, pick up the liquid for Y, and 1 drop on Y. To be very careful not to spread it too much. And see. Here we go. Here I have all 4 pigments on my filter paper. The next step is to put this filter paper on water. Once you have your paper labeled and all the pigments put in the appropriate place, you're going to bend your paper slightly like this, curve it so that it can fit into the beaker. You're going to fill the beaker with water up to the first marking, and then you're going to place the filter paper right in the beaker and wait for the pigments to rise. This will happen fairly quickly. Within 5 minutes, the pigments should rise and give you a very clear indication of all the colors that are in each plant species. You can see it rising now. As the pigments go further and further up, they separate into different colors. You will need to plot your data from the paper chromatography on column 4 of the lab. You can directly indicate the colors that are shown in the paper chromatography. So you can directly say that BC shows orange and then blue, and whatever you see for X, whatever you see for Z and Y. Indicate them in the corresponding boxes. After your paper chromatography portion of the lab, you will complete a test for enzyme M. Enzyme M is an enzyme that is present in Botanicurus. And the thought is, if this enzyme is present in Botanicurus and it's present in another plant species, maybe that plant species relates to Botanicurus as they share common enzymes. Now, the scientists cannot directly look and test for curol because that's not always possible. They are able to test for this enzyme and use this enzyme in order to find plants that may have curol. Enzyme M, very fortunately, has a bubbling reaction when added to a testing powder. In order to conduct the test for enzyme M, you will turn to page 3 on your lab report and read the portion right below test for enzyme M. These directions will tell you the purpose of the test and why you are testing for it and what you should expect to see. Please read that and then complete the corresponding question that asks you the importance of enzyme M. Once you have read these brief directions and you've responded to the question, you will move on to the test. In order to complete the test powder is placed in each 4 wells of your tray. You can label them Botanicurus XYZ based on where you plan to put the plant extracts in. So for Botanicurus, I will use my pipette, take some plant extracts from Botanicurus and drop it into the Botanicurus well. Place it in the well indicated for Botanicurus and observe what happens. What I see is a bubbling action. This bubbling action indicates that enzyme M is present in Botanicurus. We'll take a different pipette and try another one. This is X. I do not observe a bubbling action. After you've completed the test for enzyme M, step 9 is to complete gel electrophoresis. Gel electrophoresis is typically a test conducted in lab using sophisticated soft technology and gels and DNA segments. Gel electrophoresis cuts DNA segments to compare those segments in paternity tests or other comparative DNA tests. You will conduct a simulation of this test using a paper representation of DNA. So here we have a DNA segment for Botanicurus, and I will act as the enzyme and cut this DNA segment in the following way. Every time I see a sequence that says CCGG, I will place a slash in between the two C's and the two G's. So I scan, I see CCGG, I place a slash. Continue on, scan, CCGG, I place a slash. So I've cut the segments for Botanicurus. Now I will cut the segments for species X. I will scan, see CCGG, and place a slash. Please do this now to all of the DNA segments for the rest of the paper. Once you have cut your DNA segment, complete question number 3 that asks you what gel electrophoresis is right over here. And then move on to the next page to describe how does gel electrophoresis work. Once you have done these two, now you are ready to turn to the last page of your lab in order to conduct the gel electrophoresis. Now, cutting the DNA is only step 1 of gel electrophoresis. The second step is to separate these DNA segments based on their sizes. So we look at this chart. We have DNA sizes over here, and we have the 4 species that we're comparing over here. We will take our DNA strand. We will do 1 species at a time so as not to mix the DNA strands. So I'm going to take my first one, and I'm going to cut out the sequence up to the slash. I'm going to cut it thin so it's nice and neat. And then I will count nitrogenous bases in this DNA strand. So there are 1, 2, 3, 4, 5 bases. And it's Botanicurus. So I will put this under Botanicurus and I will put it next to number 5 because there are 5 bases and it's Botanicurus. And I will glue this on in this space over here. After that is done, I will do the next one. So I cut. And I count the number of bases. 3, 4, 5, 6, 7, 8, 9, 10, 11. Then I put it in the corresponding spot. I do that for all species. And when I am done, I will be able to complete this column, the last column. We have skipped this column, we will do this at the end, but now I am able to do this gel electrophoresis comparison. A short way to fill out this table is simply to indicate what DNA segments I have for each. So for Botanicurus, I've had so far 5, and I had 11. So I will write 5, 11, and I will indicate which other species is closest to Botanicurus. After you are completed with the gel electrophoresis, you can move on to the last step of your lab. You are almost done. To complete this, you must turn to page 7. Page 7 will ask you to complete a DNA analysis. You will analyze the DNA codes to see which species is the most similar to Botanicurus. The first thing you must do is take the DNA sequence indicated here and convert it into an mRNA sequence. The corresponding nitrogenous base for a C in mRNA is G. For an A, it's U because there are no T's in mRNA. For C, it's G. Fill out the entire row for each one using this method. Once you are done filling out the mRNA, you can use this chart in your guided notes sheets that show the 20 possible amino acids in order to complete the sequence of amino acids. So the first segment that I have over here, the first codon, is GUG. I will use my chart to find that. G is the first letter, U is the second letter, so it must be here. GUG codes for the amino acid val, so I will put VAL. I will complete this for the entire, using the mRNA sequence, not the DNA sequence, but the mRNA sequence for the entire row, for all of them. Once you are completed with this, turn to page 8 and compare the amino acid sequence of X, Y, Z to indicate how many amino acids are different in each one of these as opposed to Britannicurus. And that concludes the lab. Once you have completed steps 1 through 10 in your guided notes, and you have completed all of the tests on page 8, you will now answer the questions on page 5. You will answer the questions on page 6, and read the brief passage on page 7, answer the rest, and then you are done completely with this test.
The state lab
Is frequently on questions in the Regents exam, so it's an important one to not only complete, but to really know what you're doing while you're completing it. Thank you. At this point, we are going to observe the plant stems, cross sections of the plant stems under a microscope. So we take Britannicurus first, slide it into our microscope beneath the microscope clips, right underneath the clips. We identify, some of the slides will have a green tint to them, so we identify where it is, and we want to put that spot in the middle of the microscope so that we can find it very easily. And then we want to start with the lowest possible magnification. In my microscope, it's 4. That's the lowest. It's usually the smallest objective. I click that into place directly over my slide. And then I'm going to use the coarse adjustment, which is the big knob over here, the coarse adjustment, until I can identify it, center it, and focus on it. Once I've done that, and it's visible, I am going to now change the magnification slightly by making it, by moving the objective to the next highest magnification. Now that it's at a higher magnification, I can use the fine adjustment, which is the long knob, adjust it until it's perfect and clear. And once I have it perfect and clear, I can make it bigger yet again. And then I can magnify it until it's clear, move the fine adjustment until it's clear, and then I should be able to see it, the plant in the highest possible magnification. That's how slides are used in the microscope, and then you will do the same process for X, Y, Z. Once you are done, your data from this goes on underneath the microscope column on page 8. You can draw directly what you see inside the microscope onto that column for each one of these, or you can write what you see.